Good morning, Zhongwei.
Good morning, Jennifer, and good evening to everyone here from the US, and good morning, Singapore. Jennifer, this is the first webinar series for our great initiative, Making Reproductive Longevity a Reality. And I would just like to introduce you to our audience here as my co-host, lovely co-host from the Buck Institute. And I would like you to share on what Buck Institute is trying to do for reproductive longevity and equality.
Absolutely. Thanks, Zhongwei. And to everybody in the audience, if you want to ask a question, there's a Q&A section, and we'll ask questions and answer them at the end. I'm really excited to be able to share with you and everyone our research efforts at the Buck Institute to facilitate research around female reproductive aging. So this is a photo of the Buck. We are located in Novato, California, about 20 miles north of San Francisco. And we are the first freestanding research institute to focus on trying to understand the biology of aging. And recently we started an initiative with the help of the Bia Echo Foundation to try to understand why females go through reproductive decline in midlife. So everyone in the audience will be familiar with this graph. We all know that there are a series of stereotyped changes that start happening in midlife, which we call aging, where we get sort of a decline across all of our tissues. And none of this is a surprise to anyone. But there's an exception to this, and that is female fertility. So female reproductive organs start to decline at a much faster rate than the rest of the body. And essentially, they fall off of this curve. And so that notable exception is really about ovaries. And ovaries show signs of aging decades before other tissues. In fact, when women are at their prime, in their 20s and early 30s, their ovaries are already starting to become geriatric. So we want to understand what is it about ovaries that cause them to age prematurely. And in doing so, we hope that we'll learn something about aging in the rest of the body. But this is really about more than just having babies and fertility. So it turns out that, beyond reproduction, the end of fertility sets off a cascade of really negative health effects in a woman's body. And so going through menopause in midlife has profound effects on every aspect of a woman's body. So it increases a woman's risk exponentially of things like heart disease, stroke, arthritis, osteoporosis. It changes cognitive function and immune function. I could go on and on. But essentially, if you think about what that means, it truly is the worst thing that could happen to a woman's body. And while the downstream consequences are clear, we don't really understand why this happens. And from our perspective, every aspect of a woman's life is influenced by the fact that she's going to go through this reproductive decline in midlife, separately from her ability to conceive. Her overall health, her career choices, everything about her life is impacted by this. And now that we've actually done some great work to extend human longevity overall, what this means in practice is that soon women will be living more of their lives after menopause than before. And so this is a societal problem, and this is something that we think deserves a lot more attention than it's getting. So one other really notable thing that I think people don't necessarily realize is that a woman's reproductive span, so the age at which she goes through menopause, is also correlated with her overall lifespan, which means that, if she goes through menopause early, she has an increased risk of dying early. But why women go through this precipitous decline, we have absolutely no idea. And for the rest of the audience, for the men in the audience, the fact that women's reproductive span is correlated with their lifespan is also important for you, because brothers of women who go through menopause later also tend to live longer. So there's clearly a genetic component. It's a very complex process, and there are lots of things to think about that might influence reproductive aging. But it's clear that there are mechanisms operating that are important for both men and women.
Yeah, but Jennifer, I think that is a very, very important point that you've just put up, that when I care for women in my practice as well, I also have to emphasize to the men and their family that, whatever the woman's going through, it not only affect the poor woman in front of me, but also her brothers, her family, and important men in their lives. Thank you very much for bringing this up.
You're welcome. And this is a chart that every woman in America, at least, is shown and made to believe is an undeniable truth. So what you're looking at is the number of eggs that a woman has throughout her life. And you can see that, a lot of her egg capacity has declined before she even starts to use her reproductive organs. But what I'll say about this is that we really want to reframe the narrative. So while this is an average, on average, this is true. At the level of the individual, there's a lot of stochasticity that occurs. And, it's not necessarily this linear path. Lots of women in their 50s go through fertile cycles, and many women in their 20s and 30s go through periods of infertility. And it's cyclical and it's complicated. And it might be maybe one of the most multivariate signatures in human health. And so we want to treat it that way. And this is why basic research in this area is really critical. Oops. So coming back to what we're doing at the Buck Institute, we've started an initiative around trying to facilitate research in this area. So the Global Consortium for Reproductive Longevity and Equality is just what it sounds like. It's a global effort. And our goal is really to facilitate and translate scientific discoveries into products that might actually improve women's reproductive health and lead to, hopefully, balancing the scales in terms of equality. So the GCRLE is uniting two disciplines, reproductive science on the one hand and geroscience on the other. And part of what we're trying to do is to build an ecosystem that will allow things to go quicker. So let's see. There's really, sorry about that. There's really three areas that we're focusing on. So one is clearly research funding. So there's been very little work done to try to understand what's happening in a woman's body as she ages, and particularly to her reproductive organs. We don't understand what makes a good egg. We don't know why women go through this reproductive decline in midlife. It turns out that as a species, humans are very strange. There's almost no other species that go through menopause, and so it's clearly not necessary. So we've given away grants to support research in this area. We're also trying to facilitate bringing in scientists from outside to work on this problem. So what that meant for us was building a core facility to work on reproductive biology at the Buck Institute, so that if I'm a scientist working on something else, but I have some ideas or I think that I have something to contribute in this area, we can lower the barrier to entry. We're also building an intellectual network that we hope will have a global reach. And the goal here is to get people who don't normally talk to each other to communicate so that things can move faster. So in August 2020, we gave away our first grants to 22 really wonderful scientists. And you can read about their work and the things that they're doing on gcrle.org or on the Buck Institute website. And the purpose of this webinar series is really to highlight the work that each of our grantees is doing and to get you and the audience excited about why we want to do this and to take part in becoming part of our network. So I don't expect you to read all of the text on this slide, but I just wanted to show an overview of some of the projects, some of the areas of biology that are covered by the science that our grantees are doing. It really spans the gamut from really basic science all the way up to really translational studies. And we hope that you'll be excited to hear what people are working on. Last but not least, I wanted to invite everyone who's listening to go to gcrle.org and sign up for our network. So we're doing announcements there. We are hopefully starting dialogue around papers that are coming out and companies that are being started. We really want this to be the center of the universe, a hub for talking about what's happening with respect to female reproductive aging research. And we wrote a white paper describing what we're doing, which you can download on the Buck Institute website at buckinstitute.org/GCRLE. And I think at the end of the day, our goal is to try to extend female reproductive span so that we can balance those scales and hopefully bring a little bit of equality to the world. So that's all I have to say.
Thank you, Jennifer. I think it's amazing. And I think the audience around us will now have lots of questions, but please hold those questions and do type in our chat box. Jennifer, you have brought up so many important facts, that narrative that you've been telling me about. I seem to be doing this every single day in my clinics. And this typical case whereby, I see a 44-year-old woman, who's trying to tell me, I decided that maybe I want to have a baby, maybe, and I did a test for her to try to see how much ovarian reserve there is. And there's only one clinical marker that I say is not the best, but probably it's the only one that I could really rely on at this point in time, which is the anti-Mullerian hormone. And it comes to really, really low. And I have to tell the woman the same thing, that I'm sorry that, probably, your reproductive potential is, not great. And perhaps, you at this age of 44, you might be experiencing menopause soon. So, we have to reconsider certain options, and maybe IVF is really not even that fantastic an outcome for you. I need to change this narrative, and I want to get the science out to find out more. What do you think, Jennifer? Who do you think?
Changing the narrative? Yeah, I mean, that's the goal.
We need to change the narrative, isn't it?
And it is true, right, that AMH is a terrible biomarker for reproductive span, but it's the best thing that we have right now, correct?
For now, that's what I'm saying.
For now, right. Well, then I guess that leads me into introducing our speaker. So we're going to share with you every week 2 talks, this week just one, but 2 talks from our grantees to try to highlight the research that they're doing. And so I'm really pleased and so happy to introduce Coleen Murphy as our first speaker. So Dr. Coleen Murphy is a professor of genetics and molecular biology at Princeton University. She's also an expert on female reproductive aging. And she happens to also be an inaugural grantee from the GCRLE. She has a Senior Scholar Award. And Coleen has won so many awards that I'm actually not going to read them all. But she is an incredibly creative scientist. And one of the things that I think sets her apart is that she spends a lot of time mentoring. In science and in medicine too, it's so important to have strong mentors. And I consider Coleen to be an amazing mentor for me and for a lot of other scientists in the same boat. So Coleen is working on a project to identify biomarkers that would actually replace AMH and potentially go much further than that. In order to find diagnostics that would allow scientists and clinicians to figure out what makes a good egg, what is it, and how many good eggs does a woman have, how long is her reproductive span. So I'm going to turn it over to you, Coleen. Welcome and thank you.
Thanks, Jennifer. That's a kind of embarrassing introduction, but thank you. So I'm really glad to be joining you. Unfortunately, I'm not in a fancy seminar room. I'm in my house in New Jersey. So hopefully you will be able to, let's see if I can share my screen. All right, so what I want to tell you guys about today is the project that the GCRLE is generously funding for us. And so it's too early for us to give you results, but I'm going to tell you about the work that we're doing and sort of the mindset that we have going into the project. All right, so as you've heard now a couple times, the odds of a woman being able to have children declines as she ages. And this, of course, this starts to be a problem in a woman's mid to late 30s. With the rise in infertility and events like miscarriage that are due to chromosomal abnormalities. And as Jennifer just pointed out, there's actually a difference here between when a woman might go through menopause here. The average age in the United States is 51 years, but problems can start to occur almost 15 years before that. So I think there's a set of issues that center around a woman's health and menopause. And in time, actually, though, it's almost completely separated from these issues of infertility. And as was already pointed out, like this correlates with a decline in egg quality, but of course, egg number, but actually egg quality is really the important determinant of whether a woman can have children or not. And we see this in the news all the time. We're sort of just bombarded with this idea that women's time is running out, and it's actually quite stressful. So even if you weren't thinking about this before, like, it's almost hard to avoid in the popular press. And so the problem is that we have these averages, right? We have this average information for the population, but you as an individual, there's no great way to know this in advance. I just had to guess when I have my kids, and you can't walk into a doctor's office and say, I'm a 30-year-old woman, how much longer can I have kids? And of course, you can get things like you can have the number of eggs measured, and as was referred, hormone levels like AMH can be measured, but the time scale that this gives you information about is not great. So in order for women to have really like an equal, fair lot in life, it would be nice to be able to make plans. And the information we have doesn't give us that information on a scale of years, it's only on months at best, if at all. And so right now there's no non-invasive, long-term, like years-long biological tests to assess your individual stage of reproductive aging. And this is the problem that we would like to address in our project. All right, so what we're thinking is that the way to go about this is to try to get a lot of data, so large data set analysis on clinical samples. So we're going to get into what kind of clinical samples would be useful for this, so that we can actually understand this middle part. You see, there's this real decline between 30 and 39, and this increase in miscarriage rates, right? So we're really interested in what's going on in the mid-30s. All right, so we've decided there's two ways to address this. I'm going to tell you about both of the projects. The first is this idea of, can you look for genes that are in common between blood and aging reproductive tissues? All right, to give credit where credit's due, Yuxin Su at Columbia University shared these data with me. She and I are really close collaborators on many projects. And so she asked the question, does gene expression in the aging blood and ovary correlate? All right, so the data was taken from the GTEC study. So this is a large consortium. It actually keeps getting built. There's more and more data all the time. This is from the version 8 from 2019. So the data in the study are from individual people. It's all post-mortem pretty much, except for a couple tissue lines. And what happens is these donors give samples for their all these different tissues. So we have 54 different tissue types. And of course, we have no control over the ages that go into this. I'll get into that in a second. And so what we're interested in for the purpose of this study is from these donors, could we look at ovary and whole blood, so PBMCs? Now, I mentioned ages. So of course, when people die, that's not like a totally normal distribution. And what you can see here, if you just look at the two tissues we're looking at, ovary and whole blood, there's more tissues or samples available, or these gene expression samples available in the 50s to 70s, okay? And if you recall, what I'm really interested in is like in the mid 30s. Nevertheless, so Yuxin and her team went through this. And so in the 20 to 50 year age range, they were able to get 94 samples, but found no real significant genes of change in the whole blood, and same problem for ovary. When they expanded this to include the 60-year-olds, they were able to get a lot more samples. So not surprisingly, they were able to find some genes that change with age, and some fraction of those genes overlapped between the blood and the ovary. However, this is not the greatest type of analysis. We see some correlation, but it's not exactly what we're looking for. But here are the data. So 245 aging genes were present in both blood and ovary. Okay, so as you kind of gather from that, I'm not really super satisfied with that because it doesn't really give me a strong sense of what's going on in the age range that I'm interested in. Maybe someday GTEx will have a lot more samples in that range. But for right now, I want to sort of go through the thought process that we went to thinking about a totally different approach, right? And that's really to look for, use an unbiased approach to look just for genes that change in that same age window that may indicate a systemic change, all right? All right, so actually Jennifer referred to this a little bit, and it's kind of interesting to bring it up. So Tom Pearls and others had a paper back in the late 90s where they looked at women who were born in 1896, and they compared centenarians with women who had died by the year 1969. And what they looked at was the difference in when these women were able to have kids. And of course, this is, independent of any artificial reproductive technology. So this is really just when they were still able to have children. And as you can see, centenarians were much more likely to be able to have children after the age of 40. Okay, so that's our first clue that there might be something going on. And there's been other studies since using much larger cohorts. This study from Sun et al. And also Tom Pearls used the long life family study data. So again, these are a large group of people so basically what they tracked was longevity and also when they were able to have kids. So there's a lot of information here. I'm just giving you information about the study. You can look at their paper. But the real story here is that women who had their last child after the age of 33 were twice as likely to be in the top sort of 5% of survival, living over the age of 95, compared to women who hadn't had their kids that late. Excuse me. All right. So we're getting this sense that there's some connection between late reproduction and longevity. And here's another one from the Uri Laufey's group, again, suggesting there's a connection between late births and maternal longevity. All right. It's nice that this paper, and actually the next one, they're trying to say that women who live a long time could have kids late. They're not trying to suggest that there's a causal relationship, which is interesting because that always comes up in the news and the newspapers always get this flipped. And here's another paper saying the same kind of thing, cautioning people that having kids late is not how you can live a long time. So that's not the take-home message. The point is that women who live a long time are more likely to have kids late. So maybe it's a biomarker of longevity. And the last paper I wanted to mention before telling you how we think about this is there was a study, again, from Uri Laufey's group, where they were interested in looking at what happens with women who can have kids really late naturally. So these were, it's a small, very small sample size. So these were only 8 women who were over the age of 45, but they'd been seeing this in their clinic for a while. And so they just asked what's different in their blood. So this was back in the days of microarrays. And so they did some microarrays on that and reported the results. And they did find some differences between the gene expression in the blood of the women who could have kids really late and those who couldn't. All right, so how did I get into this? I'm not going to talk about C. elegans, even though that's my real main area of expertise. But I will say that we figured out we could use C. elegans as a model for human reproduction because you'll notice both worms and women have a very long post-reproductive lifespan. And we did a lot of studies that I'm not going to go through, but what we discovered is that both worms and women, their reproduction is limited by oocyte quality, and oocyte quality declines with age, and the same genes change with age in both worms and women, which is surprising considering these are worms, those are women, and the oocytes that have been sitting there in women for decades, and for worms it's only days. But basically what takes to be a good egg in a worm is the same as what it needs to be in women, okay? So there's some similarity. And the reason I'm bringing this up is because we already know a way in C. elegans to extend reproductive span. So this well-known gene called DAF2, so this mutant in the insulin receptor, not only do they live a long time, but they also have a very long reproductive span, almost twice as long. All right. And those mutants retain their egg quality with age. They don't have that same change with age in their oocytes. All right. So again, we're seeing something systemic that connects longevity with reproduction. All right. So I mentioned all these ideas of like extended fertility, extended lifespan may be linked. What I didn't tell you is when we dug down into the Gilchinsky data, in that study, we discovered that in their data, they had several genes in the insulin signaling pathway that are downregulated in those women who can have kids really late, all right? So this was our thinking. This is what we knew going into this project. And so the idea is, could we find a set of genes that change not in the woman's ovaries or something that's really hard to get to, but instead just in blood? Is there a systemic change that we could track? Now, this is a model. This is not real data. Well, it's real data, but it's not our real data. This is from Bruce Yanker's lab when they were looking at brain changes with age. But this is a model of the kind of thing we'd like to look at. And that is, could we look at gene expression changes in blood and be able to say, these are the most highly changed genes with age in blood within this window that we're interested in, from 20 to 50, right? So if we take that and we have the genes that are most up and most down with age, and we can see where women in their mid-30s are, could we build basically a clock to look at reproductive aging? And so I'm going to show you our preliminary data. This is nowhere near finished, but this is what we tried to build a data model just off of a set of about 50 samples. So this was a collaboration with just a clinic near us, an OBGYN clinic, and we took blood from volunteers at this clinic. It was mostly nurses, so it was kind of interesting. So we have a 51-year-old nurse who got excited about giving us blood here. So we have a range of ages from 25 to 51. And as we really wanted to have more samples within the mid to 30s to 40 age range. All right, so it's sparse, but it's got the right kind of distribution. And so the first thing we did was just do gene expression profiling. So we did RNA-seq, and we looked for the genes that were differentially expressed. And in this, even though this was just a small set, we were still able to find 36 genes that are significantly different when we divided the groups up into these quartiles of ages, okay? So then we were able to take the data set and ask a different question, which is, how do women look, these profiles look using these genes? Okay, so what I'm showing you here now, you'll see on the right, the age group. So this is the age of the women from 24 to 51. And these are samples. And what you can see is that if we look at the young, which is this green top line, we've got middle age in the orange and the older is in the periwinkle. We see largely, two sets, big blocks, right? You can see this sort of orangish block for young over here and the blue with age, right? And so this tells us that this kind of works. So even though it's a small data set, that we can order our samples by age. And I hope what you can see right away is something popping out at you. If you look at this, there's at least one, maybe two 31-year-old samples here that actually skew older. They better match the older ages, okay? So this is exactly the kind of information we're looking for. We're looking for, if you could take blood, could you ask, instead of chronological age, could you ask biological age? And I would say that this sample is actually on the older side, even though the patient's only 31. Okay, so we're in the process of building a better set, and I'll tell you about that more. But let me just tell you what we can imagine using this for. So that 31-year-old patient, if you were a doctor of that patient and you saw this data, you might, say, well, you better match a 39-year-old. How would that change how you plan your life? That's the real question. On the other hand, you might have a patient who's older than that, but actually has a really young profile. And that patient may still be a good candidate for something like IVF. Even though in some countries that's no longer they wouldn't necessarily support that. So this could give you a good reason to have that patient go ahead through that process. We also saw in our clinical data, I didn't show it because we moved these samples that they looked like this, but we could actually see samples that looked like they had PCOS just based on our blood samples. And then the final thing that I'm really interested in is, we've shown this for patients, but another thing we could do is build the same kind of clock in mice and use that to test things like reproductive therapeutics, because that's something we wouldn't want to do in humans yet, but this would give us a way of asking whether things really slow down. So I mentioned we were scaling this up, and actually, if it hadn't been for the snowstorm today here in New Jersey, we would have actually received these samples. But we're collaborating with Zeb Williams at Columbia University Fertility Center, and they are basically scaling up this whole thing for us. They're collecting blood samples from their clinic from women aged 20 to 50. And I should have mentioned before, we do a whole survey of health information, so we can do things like see if this patient might actually be pre-PCOS. And then once we get the samples, we're going to, of course, carry out transcriptional analysis, but we're also adding DNA methylation to see if that will also give us some information as well. And so that's the idea behind what we're doing. We really want to use this expanded sample set so that we can build this better clock for reproductive aging and maybe get away from, as we heard at the beginning, things like AMH, which may not give you the window that you need in order to make decisions, but instead, like, every year you could go in, a woman could take have a blood sample taken and then say, like, does my chronological age match my biological age or does it not? And can we make decisions based on that information? All right, so with that, that's all I have for you today. I just want to thank the very important people who are collaborating with us. So Yushin, I mentioned, she's a really close collaborator on many projects, and she did the GTEx analysis that I mentioned. Zeb Williams is very nicely collecting the samples that we need to be able to do this analysis. And all the work in my lab is really spearheaded by Rachel Koletsky. She's a fantastic scientist, and she's been figuring out how we can best do all the RNA-seq analysis. And of course, I want to thank the GCRLE for their support of the project as well. All right, so with that, I would be very happy to take any questions. And I'm going to stop sharing my screen so I can see you better.
Thank you, Coleen.
It's an amazing talk, Coleen, and I have so many questions myself, but I can't ask my questions yet because I have to answer, I have to ask the questions that the attendees have. And I think the first question I have for you, which is really exciting, is what do you think of the biomarkers you are going to discover for women's reproductive span? Will it also predict her health span and her lifespan?
So that's a great question. I think that they will. The real question will be, you know, other people have been doing, I think, kind of a similar idea. We had this idea a long time ago. We've seen more and more experiments where people are taking like similar blood samples all the way up through centenarians. So that's a different gene set. But I do think that, you know, I think that all of these things are good indicators of what will be health span. And this correlation between centenarians and late reproduction, I think, is pretty undeniable at this point. So I think it would be surprising if we didn't find biomarkers that correlated between reproductive span and lifespan.
Right. I think that's excellent. And we have another question from one of our attendees. I think this is a very important question. I like that question. I think, can general healthy aging interventions be applied in healthy reproductive aging intervention? That's the first part. The second part, should healthy reproductive aging start earlier than even healthy aging intervention, considering that the ovary, ages at a faster rate?
Okay, so that's a great question. All right, so I should have mentioned one of the very early things that sort of got me interested in this project was that I gave a faculty, at our department, we give faculty talks about our work. And I think I just started my lab and I was interested in reproductive aging. I was talking about worms. And Becky Burdon came up to me later and she said, she was talking about PCOS patients and the fact that many of them are actually prescribed metformin. And so, it would be interesting. And sometimes people actually regain fertility. So I don't know all the things that you know, but it does seem that there's this correlation, and maybe some of the things that we think of as like aging interventions, but we only think of like applying to older people might be really useful. But yeah, and we'll know, if we can use this clock, that would be one way we could know this, right? You could actually do this test and do this intervention for like a year, and then see if you test younger than after that.
Right. Excellent.
Yeah. Can I add to that, actually?
Yes,
one of the grantees that we funded in this first round, another senior scholar, Mary Zielinski at OHSU, she's actually doing this. She's testing what is a healthy aging intervention in a preclinical model to see whether or not it affects reproductive aging.
I should ask her for her blood samples. Yeah.
Actually, yeah. For anyone on the call who's interested in tissue samples, they're going to be available to the community, actually.
Awesome.
Yeah. I'll go ahead and ask another question from one of our, there's a lot of questions. And this is actually comes on the heels of what you were just talking about, Coleen. How does the correlation between genes and age relate with the concept of metabolic age? Lots of clocks.
Well, no, this, of course, all this stuff, it's a little tragic that we're just now getting samples, and in a year we'll be able to answer all these questions much better. But so right now I don't have anything that's definite to say about that. But given all the correlations between metabolic aging and lifespan, and it would be surprising if it didn't work out. But
In young women, so, the correlation between healthy aging and reproductive span is really clear, even in very young women, right? So young women who have issues in their reproductive organs, like women with PCOS, for example, are much more likely to develop metabolic diseases. Yeah.
That's right.
So there just seems to be at least a correlation there, if not a causation.
Yeah, absolutely. And that's another question. I'm not sure if Coleen has any insight. Is that they were asking you what preliminary findings on your gene expression study with a varying age? Which gene ontology pathways does most of these genes belong to? I mean, it'd be quite interesting because
sorry.
leading on to metabolic, disorders and PCOS and aging, I'm sorry, I want to know the answers as well.
Okay. So again, I'll have to check back in a year because 36 genes is not really enough to build anything really intelligent on. There are we saw a few things with the insulin pathway, but really in order to do good gene ontology analysis, you need many more genes to be able to say that. So this is what I told Jennifer before this. I was a little worried about not being able to actually answer with true data.
No, that's okay. I think it's great to just pose the questions and to think about what.
We'll definitely be looking for that, right? So once we get a good gene set, I don't think it'll be random, but we don't have a good, right now, I don't want to say that we found something that we haven't, been able to get enough samples to say it.
Right.
Another question from a grantee, from an audience member is, do you know if there's research focusing on microbiome, particularly vaginal microbiome, in relation to reproductive aging?
Okay. So I'm very interested in the microbiome. I don't know, maybe Jennifer would know this better than I would, since you guys saw the grant applications, but I don't have an answer for that. We haven't incorporated that in our study because, of course, it'd be a much more involved than just taking a blood sample. But I think it's definitely worth looking into later, because the trick with microbiome studies is, of course, cause and effect. Right? It's you could imagine that a patient who already has a metabolic shift in some way might actually be a different host for a microbiome. And it may not be causative. So there might be a correlation, but we wouldn't know yet whether it's cause and effect. But it's a good idea for later. Yeah.
It's a great idea. And there are several startup companies actually working in this space. But there's also, I know of at least one researcher at Columbia University, so where your collaborator is, Coleen. Tal Coram is working diligently on looking at vaginal microbiome across a very large patient sample. So that'll be really exciting to see what happens there.
Yeah, and actually to chip in from the fertility studies that have been ongoing, the vagina microbiome actually has been looked at, and even the endometrial microbiome has been linked to implantation and conception rates as well. So I think that field is getting bigger and bigger, and reproductive aging could be one of these that we need to sort of examine further. And I think that'll be quite exciting in terms of clinical regimens that I can offer women, especially when Coleen was mentioning about giving the prognosis based on the age of the women, not just age. Because I have this question, which I think I should be answering it instead of Coleen, because it's asking
the
about
AMH question.
Yeah, AMH. What AMH figure would be a good range for women with age 36 years old? And I'm so sad to say that I can only tell you what the laboratory ranges tell me, and it's not really age related. It's just telling me whether it's good ovarian reserves or poor ovarian reserves. And if it falls in a good range, that's it. I can just tell you probably have enough eggs, but I can never tell the quality of the eggs. So what Coleen, you have actually suggested is exciting to me, because now I can actually prognosticate someone, like you're 31, but it doesn't mean that you can wait for another 2 years. I'm sorry, because science is saying something else. Yeah. So what are your thoughts?
I'm hoping it would be, and we're hoping that we can make it into something that's cheap enough that a clinician would feel comfortable just throwing that in with the rest of the blood panel when you have a patient do an annual exam. That would be the goal. Not to make it something extraordinary so that women only wait until they're thinking about having kids and then having trouble, but it would be something like just annually so that you'd be able to track it your whole life.
Great. I could imagine it being applied to really to careful studies to look at. I mean, ovarian aging is influenced by so many different things, right?
Yeah.
Genetics for sure, but then also environment and underlying conditions and environmental factors. I mean, there's so many things that influence it, and I could imagine doing really careful studies to see how each one of those things individually affects. Yeah.
And if you could reverse it, right? That would be so wonderful.
You could slow it down, even just slow it down a little bit.
Yeah, that's right. Yeah. You could have
an extra 5
over and over
years. Yeah, exactly. There's a question which I think we could probably all answer. I'm curious, how good will the mouse or the rat as a model for female reproductive aging be? And I will say, having looked at all of the grant applications, one of the things that we are sorely in need of in this field is new model systems. They're really, there are models out there, and none of them is perfect. And I think this is one of the reasons that we want to attract people from outside the field to come in and really get creative, is because mice and rats are great for some things and not for others. Did you want to add to that, Coleen?
I'm going to have a very weird comment because, since we use C. elegans, I've always been struck by the fact that that even the same genes come up. So we had this crazy finding with the DAF2 worms that when we looked at oocytes with age, we actually noticed that there were genes that were downregulated in the good oocytes. And they turned out to all encode the gene for encode cathepsins, okay? And so these cathepsins, we were like, well, that's crazy. Like, what if we just throw cathepsin inhibitors on these? And it turned out to extend reproductive span. And we were feeling really clever until we looked more into the literature and discovered that people were interested in bovine reproduction, actually already knew this. They already knew that the genes for cathepsin were actually a good bio. Like if they're up, that meant that they shouldn't, breed those cows. And so I think that there's probably more, the model, like maybe we need to be careful, but some of the really big lessons may actually be okay across a bunch of different models.
Yeah.
That's my 2 cents.
Yeah, I agree. There's a lot of similarities. There are some things though that really that are not recapitulated by the models
and
we have.
I'd be happy to say that. That's true.
Actually, we have a question from Mary Zielinski, whose work I just referred to, and who is actually going to be a speaker later in this webinar series. So tune in for that. She asks, do you think there's an equivalent to Horvath's clock in relation to reproductive aging?
That's it's a great question. So for those of you who don't know, Horvath's clock is the DNA methylation. And so, and there's a really nice paper by Morgan Levine where she actually used the Horvath clock to actually look at the acceleration in aging that happens with reproduction. It's a beautiful paper because they were able to really show that menopause itself actually causes an acceleration in aging, which is terrifying. And so but of course, what they were looking at is menopause. We are going to be doing the same kind of analysis with our samples. So I do think that I don't know yet, but that's our hope. That's why we're doing that analysis in addition to the RNA-seq.
Right, that's really, really interesting, because that leads on to another attendee's question, is that how do you correlate the general fitness of a woman to a reproductive health? I think all of us have our opinions on this, but Coleen, I mean, your work is interesting. It's really trying to say something about that. Do you want to share something that's more, your messaging to all of us?
I just like a little bit, like I just cut out the last
Oh, sorry. Can you hear me? So yeah. So for your work, I'm just asking, can you correlate the general fitness of a woman with her reproductive health? Just based on what you're looking at.
We'll know. Okay, so we take a lot of information when we have every woman fill out a survey, so we know some general things. But yeah, so we'll know things like BMI and lots of other health factors, behaviors like smoking and things like that. So we may be able to get some of that information. I think one thing that's frustrating about reproductive aging, though, is that you can be in great shape and still have problems, right? And so, it's not really obvious. And that's why it's so important for us to study this, because there are women who are doing everything that it looks like they're doing everything right, and yet they still have these problems. So there's some unknown factor, and hopefully we'll discover that when we're looking at this.
That's exactly right. There's some biological timer, or there's some cue or a set of cues that that we don't know about. And until we actually know what is causing, what's the causative thing or things, everything that we're doing is just going to be a Band-Aid, right? It's just not going to make a difference.
That's right.
Right.
That's why we need the basic research.
Exactly. To figure it out.
Yeah, absolutely. And I think for all of us who are trying to explore this field in detail, there has been a lot of work on metformin being useful in preventing type 2 diabetes.
Yeah.
What about the data? Do we know any data with regards to reproductive longevity from metformin itself? And then do you think we'll find any modifiable factors that you identified related to reproductive aging in this case?
Yeah, so I think that's really interesting, especially because, like I mentioned before, if PCOS patients are being treated with metformin and they are able to see improvements in their fertility, then I think that's a suggestion. Right now, we have these samples that we're kind of holding out because they either are PCOS or look like PCOS. And so I don't know if we'll have enough data at the end of this to be able to say anything, but then it's interesting to know whether they're being treated or not. Right. And so I think it's a great question. I don't know the answer, but I think that there is a relationship there.
Right. I mean, there are systemic reviews and meta-analysis actually looking at a woman, who have PCOS, and we're trying to use metformin alone as a treatment versus the recommended ovulation induction agents to see whether they ovulate and, giving to live births. And so far the observation data isn't that convincing that all women with PCOS with metformin will ovulate, will have a better outcome. And it's still being more reserved for more women with insulin resistance. So I think, Coleen, your work itself, any of the genes you look at have insulin resistance or affecting the pathways could be very interesting, because that will bring us back to the metabolic kind of effects that we hope to see in these women, isn't it?
Yeah, I think if we could find a gene signature that actually correlates, and again, it would be really nice to know if we if we could develop this clock, if we could then have a metric to be able to, in addition to just sort of other features, if we could use this as a way to tell us whether these approaches are working or not, right? And maybe there's something fundamentally different between the patients where metformin had an effect and maybe didn't. Maybe from the gene signatures, we know better what that is.
Right.
Yeah.
I hope so.
I hope so too.
I'm looking forward to that so that I could change my narrative in the clinics every time.
That's the hope.
Yes.
Okay. I think we are through all of the questions. I wanted to remind everyone to sign up for next week's webinar. So we're doing this every week for the next 9 weeks, and we'll be featuring 2 different speakers each time. And next week, I'm very excited that we're going to be featuring another senior scholar, Holly Ingram from UCSF, and a Pilot Award recipient, Polina Lishko from UC Berkeley. And they're both working on trying to understand how the brain and the ovaries communicate, and how changes in sort of what's happening in your brain can influence reproductive aging. And for those of you who are in the audience, if you want to go over to gcrle.org, we can continue this conversation. We'll start a thread, and so you can ask questions. We can talk about things that maybe didn't come up here, but that will come up later. And yeah, I thank you all for attending. This was great. Thank you, Coleen.
Thank you for having me.
Yeah. Thanks, Coleen, for this very, very exciting work they're going to start. And hopefully we can find something really significant and can be immediately translatable to, to clinical utility. And I think it is amazing. And thanks, Jennifer, for introducing the next week's seminar. So it'll be a weekly affair from now onwards, and we hope to see everybody back again to continue this conversation. And Coleen has started really beautifully for us, to give us this message that the clock in a woman, is probably something that we can do something about. It's not really just what nature deems it as it is. So until next week, make reproductive longevity a reality. Thank you, everybody, and have a good day and a good evening.
I used to wonder if I ever could be loved. Never gonna live up. Should I give up? Will I ever be enough? What I didn't understand is you love me as I am. I don't don't care what anybody thinks about